So as Josh mentioned, that a significant issue for platypus is that their populations have become very separated, and we're ending up with a lot of small populations that have become isolated from all of the other populations in Victoria. And that's for any species, that's a real problem for in terms of their genetic health. So at least for platypus, it's a pretty iconic species. It's well studied. We've got, comparatively well studied. We've got a significant amount of genetic data. But how do we go about assessing the broader picture for Victoria's biodiversity for so many species where we really don't have any genetic data, or we have much less of an idea of the extent of population fragmentation? So for the last couple of years, we've undertaken a project in collaboration with the Victorian State Government to assess the genetic health of Victoria's flora and fauna overall. And I'll just give you a bit of a rundown of where we're at with that. So the fundamental problem we're facing is fragmentation of natural habitat. And that's a particular problem in Victoria for historical reasons. We are the most cleared state in Victoria. So this is what's remaining of native vegetation. 15 years ago, the rate of loss has slowed down since then, but we're still faced with this historic problem. And of course, we've put all these other barriers in place, roads, dams, fences, which all of these things contribute to populations becoming isolated. So for so many species, they're now persisting in little patches of habitat. So there are significant genetic risks associated with being in small populations. The most significant one is the risk of inbreeding. Once populations become very small, individuals will inevitably end up breeding with their relatives, and inbred species, inbred populations can be just not fit for living, or the rigors of living in the natural environment. They may persist in zoos where they're sort of spoon-fed or protected environments, but in the wild where they're facing all these different competitions from other species, that can rapidly lead to inbreeding depression. And although it might not be the initial process that causes extinction, inbreeding depression can be the final blow that sort of wipes out populations at a local level. The second problem is loss of genetic diversity. So even populations that are not so small that they are at immediate risk of inbreeding depression can be just losing genetic diversity. And genetic diversity is sort of, if you think of it as a species toolkit or their arsenal, it gives them the ability to cope with all of the different challenges that their environment throws at them. Perhaps the classic example of this is with these peppered moths in England. When the Industrial Revolution started and soot made all of the trees black, these moths were able to adapt very quickly to darkened environment and still be able to be camouflaged. So the question that we'll be facing in the future for a huge number of species is, do they have the capacity to adapt to hotter and drier climates that we're likely to face in the future? So both of those things are critically important. So to look at the broader picture for Victoria's biodiversity, we're trying to assess the extent of population fragmentation and genetic risk to all of our flora and fauna. And where we have genetic data, we can do that by directly estimating the gene flow between populations. And where we don't, we're trying to model that by estimating the dispersal capacity that organisms have, and that's sort of over their whole lifespan. And then we're also needing to estimate the effective size of those separate populations. Are they at that critically small size, or are they large enough to be likely to be self-sustaining for decades to come? And so to give you an example of how we're doing this is one of our marsupial carnivores, not quite as well known as the platypus, but still a very cute and uniquely Australian animal. It's mostly found in Victoria. They're also in Tasmania. And these are all the species records that have been collected from the ALA database. And we're trying to assess the connectivity of those. So we can do that based just on distance. So here. Lost my mouse pointer, just a little. So if we just pick a distance of 6 kilometres of what we think might be a reasonable distance that these animals can disperse, regardless of their habitat, we would end up with all of these different clusters. And that's perhaps too much it's not really a realistic representation of the fragmentation. For some species, it will be just using the distance will be a pretty good metric to use for a lot of birds and wind pollinated plants. But for a species like this, this is probably not quite realistic. But if we pick a larger dispersal value, say 20 kilometres, then realistically across open country, these animals can't really disperse that far, and we end up joining populations that probably shouldn't really be joined. So to assess this properly, we're needing to take into account the underlying landscape and the connectivity in the landscape. If we do that, then we end up with a picture that's more like this. So we've got this large population over in the sort of the east, which is likely connected, but the populations, there are several other populations in the Grampians, in the southwest, and the Otways, Wilson's Promontory. So it's likely based on this analysis that there's possibly around 15 separated populations. And then, of course, a lot of these populations are going to be at significant genetic risk because they're likely to be quite small. So, and then we also need to take into account recent events. So although we have one large population, we've probably lost a significant part of that from the recent fires. And then for some of these populations, the only records that persist, so there was a population there in Mount Napier State Park, the only records there were for more than 30 years old. So it's very likely that population's gone extinct. So we're taking that into account as well. And then trying to assess the overall pattern of genetic risk for this species. So we're using landscape models where these are available. So for some species like the striped legless lizard, there are habitat suitability models that have been developed that are particular to that species. For others, we're using a generic model where we're looking at the biodiversity values of the Victorian landscape overall. So here, dark green is representing areas of high biodiversity value, and the pale yellow is poor biodiversity value. And we're using these models to sort of where we need to model the connectivity, the gene flow across the population, and what's the likely connectivity between those populations. So we're also, where we can, we're going to try to look at the timing of the separation, where populations have become separated, whether it's only occurred recently, or whether it's possibly that possible that populations have been separated before European settlement, in which case they're likely to be more divergent and possibly more genetically valuable. But it's going to be quite challenging to do that without collecting genetic data. So in that example with the dusky antechinus, there's some reason to suspect that the Grampians population is a unique subspecies and it's been separated for a long time, but we would need genetic data to confirm that. So we can then use this information to help set conservation priorities to help conserve the genetic health of our biodiversity. So we're in the process of developing a genetic risk rating for all of these species. We're looking at 4,000 odd native species. Most of those are plants, but also all of the vertebrates that occur in Victoria. How many separated populations are there? How isolated are there? Is there any gene flow occurring at all between them? And what proportion of the individuals, so the total global population for a species, what proportion now exist in small and at-risk populations? And then we're going to gather that information, and that information can then be used to inform the priorities for the government and conservation managers. Do we go about collecting further genetic data? If so, should we prioritise that? And if we're looking to restore connectivity between populations, how should we go about doing that? So few examples would be, let's just looking at this species again, the dusky antechinus. You might, for example, look at restoring the habitat to create a habitat corridor in this southwest region. You might look at perhaps putting a connectivity tunnel underneath the Calder Freeway to connect the sort of Mount Macedon and Trentham sort of populations that are likely now isolated. You might look at collecting genetic data from the Grampians to determine how distinct that population is. And as a sort of a last resort, you might look at translocating individuals from the large population in the sort of around the Yarra Ranges or the east to a population that we know is isolated down in Wilson's Promontory. So all of these things cost money, and it's very important that the government has information that will help them prioritise the available conservation dollars so that they're spent in an effective way and we get the best bang for our buck. So that's sort of the thrust of our project, and of course we're doing that in collaboration with the state government and the Arthur Rylah Institute, which is part of that. So thanks very much for everyone for listening in.